cytology. We all know cytology is a branch of biology which deals with the study of cell. So in this segment, I'm going to cover prokaryotic cell. I am Professor Sushil Mishra. We'll do best to best study regarding prokaryotic cell. So let's start. First, we come across a cell. We know cell is nothing but a structural and functional unit of life, a basic unit of life. So cell is defined as structural and functional unit of life. Cytology is a branch of biology which deals with the study of cell. Robert Hooke in 1665. This person was the pioneer to introduce the concept of cell. He used his magnifying instrument to observe something like this, which was looking like a honeycomb. In his book Microscopy, he used the term celluli, which means empty chamber. As per Robert Hooke, cell is an empty chamber. But as the time progressed, we came to know that no, cell is not an empty chamber. But again, I told you, Robert Hooke was the pioneer. So the credit of the cell or pioneer of the cell concept, we go for Robert Hooke. It was Anton von Leeuwenhoek. He observed first living cell in 1674. In his book, The Secret of Nature, he explained first living cell. Robert Brown, a botanist, observed nucleus in the year 1831. So after Robert Hooke, there were many discoveries, but see the gap of the timing. Why the discoveries were so slow? Reason was there was no microscopy segment. Microscopy deals with the study of microscope arrangement. Now, what is microscope? An instrument which helps us to see the small structure, microscopic structure. Now, remember, for a microscope, here we have two lenses. One is referred as eyepiece, this one. Second is objective, the one which is toward the object. And if you observe closely at the objective, you will find there are three lenses. These lenses are 4x, 10x, and 40x. While the objective is 10x. So we always get a combined magnifying power of objective and eyepiece. So remember, for a microscope, two properties are very important. First is magnification. What is magnification? Means this microscope or the instrument, it will magnify the object how many times? So image size upon object size. Let us consider there is an object one of size 1 micron, but after observing in microscope, it appears to be 5 micron. That means it had been magnified for 5 times. Second is resolution. So what is resolution? It's an ability to differentiate two closely situated points. I mean, how closely situated point can be differentiated, that ability is called resolution. Now, for a compound microscope, magnification powers are 40x, 100x, and 400x. Depending upon our requirement, we use it. See, 10 for eyepiece was fixed. If we are using a combination of 4, 10 into 4, 40, or same way, 10 into 10, 100, or 10 into 40 is 400. So depending what structure we want to study, we can use the magnifying power. For compound microscope, resolution is 200 nanometers. That means if a 2 points has a gap of 200 nanometers, those can be seen as a distinct point. If it has less than 200 nanometer, they will appear 1. But see, nanometer 10 raised to minus 9. So definitely very large resolution power. Definitely these are the good resolution power. Now let's see the instrument. Yes, you could you can identify electron microscope. Yes. Now what is great about this? For electron microscope, magnification power is 1 crore time, 1 CR time. And the resolution is 0.05 nanometers. So definitely very, very high. For human eye, resolution is 100 micron. Means human eye without any aid can differentiate 2 points which are at a gap of 100 micron. Remember, this is only applicable for healthy human eye. Compound microscope was discovered by Hans and Jensen. They were father and son. Electron microscope was discovered by Knoll and Rust. Remember, MCQ had been asked on Knoll and Rust, so remember it. Anton van Leeuwenhoek, if you recollect, he was pioneer in microscopy. So the microscope which he had used was having magnifying power of 270 times, 270x. Now remember, all these discoveries which were taking place, they were very slow and scattered. The major push for the cellular study came with cell theory. Matthias Klendin, in the year 1838, he was a German botanist. His observation was plant tissue is made up of different kinds of cell, or different kinds of cell make up the plant tissue. His observation. At the same time, almost, Theodor Schwann, in the year 1839, remember the years, MCQ can be asked. He was a British geologist. He made certain observation while studying. First, cell has a thin membrane, which now called as plasma membrane. So covering of the cell, that is thin membrane, was discovered by Schwann. Presence of cell wall unique to plant cells. So he also observed that for plant cell, they have a unique covering, which is called as cell wall. So he proposed a hypothesis. The hypothesis says that bodies of plant and animal are composed of cells and its product. The two studies were independent, but later on put together to propose the cell theory. Now, when I say cell theory, first point says that all living organisms are composed of cell and its product. Cell is a structural and functional unit of life. All cells have genetic material, which is passed from a cell to next at the time of cell division. New cell always arise from pre-existing cell. Now remember, this statement was not given by Schwann and Geldin. Instead, it was later on added to the cell theory by Rudolf Virchow. His statement was omni cellulae cellula. That means new cell always arise from pre-existing cell. Exception to cell theory is viruses, viroids, and prions. We will learn them also. Viruses, you know. Viroids and prions we are going to learn in coming lectures. Types of cell. Prokaryotic cell, a cell which do not have well-organized nucleus. You can see in the diagram, it's a prokaryotic cell. It do not have well-organized nucleus. Can you recognize? It's a bacteria. Yes. Now, next we have eukaryotic cell. Eu means new. Pro means primitive. Karyon means nucleus. So when I say prokaryotic, they have primitive nucleus. When I say eukaryotic, they have new organized nucleus. So well-organized nucleus is present. See animal cell. See, this is a plant cell. Both of them, you can see they have a well organized nucleus plus many organelles, but nucleus is very distinct here, which was absent in case of prokaryotic cell. Today we are going to focus on prokaryotic cell. Pro means primitive, karyon means nucleus. So first point, prokaryotic cell, well organized nucleus is absent. They belongs to kingdom Monera. Now remember, for plant we have kingdom Plantae, for animals we have kingdom Animalia. Same way for prokaryotic cell we have kingdom Monera. Their membrane, in case of prokaryotic cell, membrane bound cell organelles are absent. Whichever organelle has a membrane, you will find that they are absent in this prokaryotic cell. For example, mitochondria, Golgi complex, endoplasmic reticulum, all are absent. Genetic material of prokaryotic cell is called as nucleoid. It is a circular DNA without histone protein. Cell wall is made up of peptidoglycan. In case of prokaryotic cell, they have cell wall and their cell wall is made up of peptidoglycan. Remember, for cell wall, there is a one exception. We'll talk about it, but generally they have cell wall. Prokaryotic cell, now onward, I will also refer to as bacteria. Bacteria, prokaryotic. Prokaryotic, bacteria, synonymous. Bacteria are of two types on the basis of their cell wall. Gram positive and gram negative. Now when I say gram positive and gram negative, it's classification of bacteria on the basis of their staining property. But remember, it's very important for us to know gram positive and gram negative bacteria, because gram negative are mostly pathogenic while gram positive are mostly non-pathogenic. So definitely very important. I have shown you a picture here. On left side, you can see a stain where you can find darkly stained bacterial cell. They are gram positive. On the right side, you can see a lightish shade, violet color. They are gram negative bacillus. If I magnify their cell wall, it looked this way different. So definitely gram positive, the first cell wall is quite thick, darkly stained. Right side, there are two distinct layers. Can you see that? Yes, and definitely it's a thin layer. It's a gram negative. Let's talk about it. The concept of staining was given by Danish bacteriologist Hans Christian Gram. Gram staining technique, how it proceeds or how we do it. First, you have to take a stain crystal violet, put it on the bacteria. After that, we put up iodine solution to fix up the first stain and then wash it with warm alcohol and water. After that, add counter stain called saffron. After doing so, if you find the bacterial cell have the dark color, then definitely it's a gram positive. If you find that they has a faint, what you can say, violet color or light pinkish color, then it is a gram negative. Now why it happens? Because of the difference in their chemical composition of cell wall. So let's try to understand what it is. So gram positive and gram negative. Now remember, for a gram positive, cell wall is smooth. Gram negative, cell wall is wavy. Thickness for a gram positive is 20 to 80 nanometer, while gram negative it's 8 to 10 nanometer, definitely very thin. Peptidoglycan has a thick layer. Peptidoglycan is thin in case of gram negative. Teichoic acid is present in case of gram positive, which is absent in case of gram negative. It's a structural component of the cell wall. Outer membrane is absent. Now, when I say outer membrane, it's nothing but a lipoprotein layer, lipid and protein layer. If you recollect here, you can see a upper layer, pinkish color. That's a lipoprotein layer. So that is absent in case of gram positive, but present in case of gram negative. Porins proteins are absent in gram positive, but present in gram negative. Gram-positive mesosome, the infolding of plasma membrane. Remember, mesosome infoldings of plasma membrane. We will talk about it, but they are prominent in gram-positive, non-prominent in gram-negative. Flagella has 2 rings in case of gram-positive, while flagella has a 4 ring in case of gram-negative. Now, just see this diagram. Can you see? On your left side, we have gram-negative, and on your right side, we have gram-positive. So if you count the ring, I show you the ring part. See, this is the ring part. You can count the numbers. And here also we have a ring. You can count the number. So here we are finding 2 rings. Here we are finding 4 rings. Clear? Great. So flagella has 2 rings in gram-positive, has 4 rings in gram-negative. Lipid component in gram-positive is very low, while lipid component in gram-negative is high because it has an outer lipoprotein layer. Next one, gram-positive, antibiotic susceptible. That means they get easily destroyed with antibiotic. Gram-negative, they show high antibiotic resistance. Gram-positive, many of them produces spore, while none of the gram-negative produces spore. Gram-positive, you will find they never show O antigen. That means O antigen is absent. In case of gram-negative, you will find they have O antigen on their surface. Now, after learning this, one question comes to your mind. Which bacteria is more harmful? If you think, you will have an answer. Gram-negative bacteria is more harmful. Reason is gram-negative, they show high antibiotic resistance. Why? Because their antigens are covered, protected because of the outer layers. So they are having high chance of protecting themselves, which is not in case of gram-positive. Comfortable? Great. Let's proceed. Now, classification of bacteria depending upon their shape. Now, in case of bacteria, if we talk about their shape, we have coccus. Coccus means spherical bacteria. Bacillus means rod-shaped bacteria. Spirillum, which is coiled. And also we have something called as vibrio. Vibrio means comma shape. You might have heard about cholera. It comes under vibrio. Now remember, spherical bacteria, if they are in cluster of 2, we call it as diplococcus. If they are in bunch, we call it as staphylococcus. If they form a chain, we call it as streptococcus. Now remember, strepto always means chain. So this is my comma shaped bacteria. We call it as vibrio. Fine. Great. So this was the classification depending upon their shape. We can also classify bacteria depending upon their origin. Archaebacteria. Archae, actually now they had been given a new domain. But remember, archae means primitive bacteria. Example for them is methanogens. You have learned biogas plant. In that methane producing bacteria, they are archaebacteria. They are structurally somehow different from normal bacteria, which we call it as eubacteria. They are considered as more evolved. Example as lactic acid bacteria, Salmonella typhi, the one which causes typhoid. We also have cyanobacteria, which are also called as blue-green algae. They are photosynthetic, perform the process of photosynthesis. Last group is actinomycetes. Remember, these are gram-positive bacteria which act as decomposer, generally found in soil. They help in the process of decomposition. Generally, decomposed process is related to fungus. They are bacteria which helps in decomposition. And mycetes word, remember, mycetes is generally related to fungus. Here, bacteria had been given a name, actinomycetes. So don't get confused. Now we will talk about the structure of a prokaryotic cell. Now this is a prokaryotic cell. If you observe, it has an outer layer which is called as capsule, then cell wall, and then plasma membrane. Together this three layer is called as cell envelope. Inside the cell envelope we have cytoplasm. Inside cytoplasm we have nucleus. We also have infoldings of plasma membrane. This word already I have used. That's called as mesosome. Generally, you find this mesosome are at the center. Other than this, we find plasmid. See on the diagram. Ribosomes, cytoplasmic inclusions, fimbriae, pilus, and flagellum. All this structure we have to study. So let's start one by one. First, I am starting with cell envelope. In cell envelope also I am starting with outer layer. The outer layer is called as glycocalyx. Glycocalyx is made up of mucopolysaccharide. Polysaccharide is carbohydrate. Remember, this is associated with mucus. We commonly find it. Mucopolysaccharide. Now remember, if glycocalyx layer is hard, then it is called as capsule. So now looking at the diagram, I had been labeled capsule, so I can easily say that it will be a hard layer. Sometimes this glycocalyx layer is sticky, then it is called as slime layer. Second component of the cell envelope is cell wall. It is made up of peptidoglycan, peptide and carbohydrate, that is protein and carbohydrate. The component are chemical component NAG, NAG, N-acetylglucosamine, NAM, N-acetylmurmic acid. Cell wall is non-living and permeable. Remember, all cell wall are non-living and permeable, responsible for shape and also to some extent provide protection. Major protection for a bacteria is with cell wall. Remember. Next we have cell envelope. Within the cell envelope now the last component is plasma membrane, which is a lipoprotein layer, lipid and protein layer, phospholipid bilayer. In that proteins are embedded. Remember, all plasma membrane, whether it's a prokaryotic or eukaryotic, chemically they are similar. Plasma membrane in case of bacteria is very important. One, all plasma membranes are living and selective permeable. So like all membrane, even this is like all plasma membrane, this is also living and selective permeable, and it gives us large number of processes. So let's see outer process. Outer process is flagella. This flagella, can you see that long hair-like structure? Yes, it is made up of a protein called flagellin. It provides rotatory movement. Remember, if you compare flagella of a prokaryotic with a flagella of eukaryotic, you will find that in case of eukaryotic, their flagella is made up of tubulin protein. While in case of prokaryotic, it is flagellin. Second, there they provide whipping movement, while here the flagella provide rotatory movement. In case of eukaryotic, ATP energy is required, while in case of prokaryotic, it is proton H plus ion are responsible for movement. Already I have told you number of rings in case of gram positive and gram negative are different. Flagella. Next outer structure is fimbriae. Fimbriae, they are short as compared to flagella, straight and helps in anchoring. They help in anchoring. Third structure is pilus. Pilus, mostly seen in gram negative, very rare in gram positive, made up of protein called pilin. They help in adhesion. Remember, in bacteria, there is a type of reproduction called as conjugation. Pilus help in formation of conjugation tube. This is mesosome. If you talk about the plasma membrane, it also gives out some inner process which are called mesosomes, infoldings of plasma membrane. Here, you can find the mesosome here. This is the mesosome. Structurally, mesosomes are made up of vesicles, tubules, and lamellae. MCQ. Vesicles, spherical bodies, tubules, tubes, and lamellae means layer. You find generally they are present at the center of the bacteria, helps in lot number of processes, which includes cell wall formation, DNA replication, division of cell. Also, they provide the site for enzyme activity. They are also site for respiration. So mesosome, very, very important site in bacteria. Mesosomes are nothing but infoldings of plasma membrane. Flagella, we were talking about it. Already this structure we have seen. Remember, they provide rotatory movement. In case of positive, we are finding different in their ring. Gram positive, gram negative, you can see the differences. Great. Now remember, depending upon the position of flagella, we have a trichus, non-flagella bacteria, monotrichus, only one flagella at one end, amphitrichus, there are flagella at each end, which are diagonally opposite, lophotrichus, a tuft of flagella at one end, peritrichus means flagella all over. So this is a classification or grouping of bacteria on the basis of flagella. Let's talk about the cytoplasm. Now remember, when you talk about cytoplasm, it's a living matrix. It's a homogeneous matrix having water, organic matter, inorganic matter. All biological activity you can consider take place at cytoplasm. But in case of prokaryotic cell, membrane-bound cell organelles are absent. That already we have discussed. There is no cyclosis. What is cyclosis? Protoplasmic streaming movement. Means like we go into wave pool, water shows waves. Like this, a eukaryotic cell, it shows a wave which is caused by certain protein fibers called as microfilament. In case of prokaryotic cells, such movement is not seen, so we say cyclosis is absent. So cyclosis is a eukaryotic feature. So in prokaryote, it is absent. Nucleoid, I told you, it's a single circular genetic DNA, which is double standard DNA. So nucleoid is a single circular genetic double standard DNA. We also call it as chromosomal DNA. It is not associated with histone protein. So histone protein is absent, but remember it shows the presence of polyamines. Amines, you have learning chemistry, having amino groups. So poly means long chain. Chain of amines, we call it as polyamines. All genetic feature is regulated by nucleoid. Basic feature, all the genetic features of the bacteria is related to its nucleoid. Next structure which we see is plasmid. See plasmid. Plasmid is an extra chromosomal circular DNA. When I say extra chromosomal, mean other than nucleoid. It's a self-duplicating structure, undergoes self-duplication, so we call it as mini chromosome. Plasmid number is variable inside a bacterial cell. They control phenotypic features, fertility, and resistance. They are dispensable. Dispensable means if we remove the plasmid, bacteria will survive. So it can live without plasmid. And because of this feature, as we progress, when we go for biotechnology, you will come to know that plasmid is a blessing for entire genetic research. So very important structure. Sometimes it fuses with nucleoid. Plasmid fuses with nucleoid, then that condition we call it as episome. So once more, plasmid are extra chromosomal circular DNA. They are self-duplicating, hence called as mini chromosome. Plasmid number inside a bacterial cell can vary. I also told you that plasmid are dispensable. That means a bacteria can survive without a plasmid. They basically control phenotypic feature, fertility, and resistance. Sometimes it fuses with nucleoid to form episome. Next structure we have to learn is ribosome. Now remember, ribosomes are non-membrane bound cell organelle. That means they are not bounded by any membrane. Present in all cell, whether it's a prokaryotic cell, whether it's a eukaryotic cell. They are present in all. In prokaryotic cell, they are of 70S type, and eukaryotic cell, they are 80S type. Now students ask me, sir, what is this S? S stands for Swedberg. It's a sedimentation coefficient. Its value is 10 raised to minus 13 second. Students find it very difficult what it is. Now please understand, when you want to learn a cell, you have to take out its component. It's not that you put your hand inside a cell and you take it out. There is a technique. What we do, we take a cell, we undergo a process, and then we centrifugate the cell. When we do the centrifugation, and then at a particular speed, its organelle starts sedimenting. So that's why they had been given this sedimentation coefficient. So ribosomes are composed of RNA and protein. There is a type of RNA called as rRNA, that is ribosomal RNA, and there are proteins. See, now if you happen to see this, you will find that this is a prokaryotic upper part and the lower one is eukaryotic. First we will talk about prokaryotic. Now any ribosome, which we call it as 70S, see here, it has two units, larger unit and a smaller unit. This is the smaller, this is the larger. If we separate out, remember in a cell they always remain separated only. They join at the time of protein formation. They join only at the time of protein synthesis with the help of magnesium. They join with the help of magnesium at the time of protein synthesis. So these two units are separated. This one is called as larger subunit. This one is called smaller subunit. Larger is 50s, smaller is 30s. Now again here student object. So 50 plus 30 is 80. Now please understand this is not mathematical addition here, because when they combine, their sedimentation coefficient becomes 70. It's not that their individual sedimentation will get added up, because that sedimentation depend upon their density, their molecular structure, their size, many factors. So remember, the 50s unit has 31 protein, 23 sRNA and 5 sRNA. R means ribosomal RNA. Same way, the smaller unit 30s has 21 proteins and 16 sRNA. So they combine at the time of protein synthesis. If you happen to see a eukaryotic cell, in them we have 80s ribosome. 80s ribosome has two units, larger one is 60s, smaller one is 40s. Again, I told you it's not a mathematical addition. 40s has 33 protein and 18s rRNA. While in case of 60s, we have 50 proteins, 5 sRNA, 5.8 sRNA, and 28 sRNA. So they get assembled into smaller and larger unit, which join at the time of protein synthesis into ribosomal unit. Ribosome, the smaller and larger unit, join with the help of magnesium at the time of protein formation. Protein factory of a cell. Remember, because protein synthesis में help करते हैं, they are called as protein factory of a cell. Now, suppose if a single mRNA has many ribosome on it, then it is called polysome or polyribosome. Polyribosome or polysome means multiple ribosome attached to single mRNA. Multiple ribosome attached to single RNA. Next, we want to talk about is cytoplasmic inclusions. Now remember, cytoplasmic inclusions are non-living component of cell. Basically, they are storage granules or storage structures. They are specialized for storage, non-living component of cell. For example, cyanophysin granules, starch granules, these are non-living component inside the cytoplasm. They help in storage. Clear this much? So now we have covered almost all the structure of a prokaryotic cell. Certain additional point we should know. Once Ames had asked gas vacuole. Remember, green sulfur bacteria, they show the presence of gas vacuole inside them. Chromatophores, these are the specialized structure seen in cyanobacteria. They have pigment and helps in photosynthesis. Chromatophores are specialized structure seen in cyanobacteria, helps in photosynthesis. Mycoplasm, if you recollect this name already I have come, they lack cell wall. If they lack cell wall, then gram staining technique do not apply for them. So remember, gram staining technique, which is used to classify gram positive and gram negative, is not useful for all bacteria. Mycoplasm are type of bacteria, they lack cell wall. They are in fact the smallest cell on entire living world. So if I have told you smallest cell, definitely the mind will come with largest cell. Ostrich egg is considered as largest cell of living world. PPLO, pleuropneumonia-like organism, comes under mycoplasm. Some places you will find this is also referred as a smallest cell of living world. It's a type of mycoplasm. Clear? So for this video, we'll talk about prokaryotic cell. We talk eukaryotic cell in coming video. So